Hello, everyone. Today we'll be discussing start site bias in DNA sequencing. This is a crucial topic in next-generation sequencing, as it can impact accuracy. So, what exactly is start site bias? Simply put, it's the uneven distribution of where sequencing reads begin, known as read start positions. This uneven distribution can affect whole genome sequencing and other applications, such as target-enriched sequencing, as well as epigenetic analysis. Ideally, we want our sequencing reads to be distributed evenly across the genome, as we see here. This ensures uniform coverage and accurate representation of the DNA sample. In this example, read distribution is uniform, showing that there is no start site bias across this region. Now, let's shift gears and talk about the causes of start site bias. Start site bias can be introduced during several places in the NGS workflow, including fragmentation of the input DNA, ligation of the NGS adapters, PCR amplification, and sequencing. In the following slides, we'll take a closer look at the impact of DNA fragmentation on start site bias. When DNA is fragmented uniformly, we get a nice even distribution of sequencing reads, and there is no start site bias from fragmentation. That's what we see here. But what happens when the fragmentation is not uniform? With non-uniform fragmentation, some regions may be more highly represented than others, such as in the pink box, and other regions might be underrepresented, as in the blue box. In this case, the sequencing data do not uniformly represent the genome. This can impact the accuracy of downstream analysis, especially in sensitive applications, such as the detection or quantification of rare variants or copy number variants. This can be especially important for applications where precise positional information is critical, such as ChIP-seq or ATAC-seq. Now that we understand the potential impact of fragmentation on start site bias, let's take a look at the different fragmentation methods currently used in the preparation of NGS libraries, and how these methods impact this metric. The first method is mechanical or acoustic fragmentation. This method uses sound waves to break up the DNA, and is often considered the gold standard due to its effectiveness and minimal bias. However, this method requires more upfront sample handling steps that can lead to sample loss, and thus is best used for abundant samples or when high throughput is not required. Another method is transposase-mediated fragmentation, or tagmentation. Here, a transposase complex fragments the DNA and adds sequencing adapters. Tagmentation requires less sample handling and is more streamlined compared to mechanical fragmentation, but it can also introduce more bias. Finally, there is enzymatic fragmentation, where nucleases randomly cleave the input DNA prior to a separate adapter ligation step. One advantage of enzymatic workflows is that they are highly streamlined compared to mechanical fragmentation and require less hands-on time compared to tagmentation. However, enzymatic fragmentation methods and reagents vary widely, which can impact cleavage uniformity. Next, let's examine how we determine the presence of start site bias in our data. We do this by looking at start site complexity charts created from the sequencing data following bioinformatic analysis. This example uses whole genome sequencing data from a bacteria that happens to have a genome consisting of approximately 50% AT pairs and 50% GC pairs. In the chart, we're looking at the frequency of nucleotides that are detected in the first 30 positions of each read. This sample was mechanically sheared prior to library prep. Here, if we look at position 10, for example, we see that all 4 nucleotides are present at this position in equal amounts, which is what we would expect for unbiased sequencing of this genome. So, in general, for each nucleotide, a smooth line at the actual nucleotide frequency with few peaks and valleys indicates low start site bias. Now let's look at the results from tagmentation. With tagmentation, we see a less tight correlation between the genome's ATCG content and the nucleotides at early positions, indicating more start site bias. Finally, the enzymatic fragmentation method used here leads to less start site bias compared to tagmentation. Now, let's take a look at human whole genome sequencing data. The nucleotide composition of the human genome is 60% AT and 40% GC. In this chart, showing results from mechanical fragmentation, you can see that the distribution of read calls reflects the composition of the genome, showing a lack of start site bias. Again, while data at all positions is informative, we'll focus on position 10 for simplicity. The data from fragmentation shows much more variation, indicating higher start site bias. This could, in turn, indicate a lack of uniformity in the overall sequencing data. Finally, looking at the results with a high-quality enzymatic fragmentation kit, we see results similar to mechanical shearing. This suggests that depending on the specific library prep workflow used, it is possible to achieve low start site bias with enzymatic fragmentation, which offers many advantages compared to mechanical methods. In summary, start site bias refers to the uneven distribution of read start positions, potentially decreasing the reliability of the NGS data. Higher levels of start site bias can decrease data quality in several ways, potentially leading to repeated sequencing runs and wasted resources. While lower amounts of start site bias lead to more reliable data and potential cost savings, and the DNA fragmentation method you use can make a difference. Thanks for listening to this Ask a Scientist video. We hope it has helped you to understand start site bias in next-generation sequencing. If you'd like to see more of our Ask a Scientist educational videos, visit the website shown on your screen.